Hello, everyone. Thank you for attending this talk. I'm so excited to talk to you today about what I think might be a novel microbiome or microbial topic to a lot of people, the wonderful world of bugs and spit. And this will be a talk about the oral microbiome's role in health and disease. So a brief overview of my talk. First, we're going to get a baseline of what the microbiome is, as well as talking about the oral microbiome and its impacts on health. And then I'm going to touch on kind of a brief case study on how we study the oral microbiome and some kind of basic concepts through my own work on the saliva microbiome. So first, what is the oral microbiome or the human microbiome? I'm sure everyone on this talk has heard about the human microbiome in some form or other, whether it's from a news story or your own work. And the most common definition that I hear and that I like to use is really simple. It's the microbes that live in and on us. But I want to dive just a little bit deeper into this definition to give you some more nuance to kind of think about these things. So the first part, the microbes that live on us. Well, what does that mean? I think a lot of us talk about, and actually this talk will focus on the bacteria. And that is certainly a large part of the microbes that are in our human microbiome. However, it is a vast community of a lot of different organisms. This can include fungi, or what's often called the microbiome, or other single cellular organisms such as the protozoum, the archaeome, or even viruses, or what's often called the virome. And this can be viruses that attack humans, but also viruses that attack bacteria as well, or bacteriophages. So these comprise the ohms, the microbes that live in and on us. But what about the in and on us part? I want to touch about where they're in and on us. I think some people often talk about the human body as a tube within a tube system. That is, we have our kind of outside big tube, and there's a smaller tube that kind of runs on the inside. And those tubes interact with the outside world. And places that interact with the outside are more likely to have microbes. So let's think about this a little bit more specifically. So that outside tube is going to be our skin. And we have a quite robust skin microbiome that varies depending on skin type. But within that inner tube, we also have our oral, our nasal microbiomes, all the way down through our intestinal system, so our esophagus, our stomach, our colon, as well as the urogenital tract. There are some exceptions to this outside exposure, but these are going to be the most robust and largely colonized microbiomes that really impact human health. This is where they're living in and on us. And while most people will often talk about the gut microbiome, which is the most diverse, it has the most number of species, and it also has the most microbes, the most biomass in it. So it truly does impact human health quite a bit. I want to kind of move this talk to a microbiome that is similarly influential and doesn't get talked about quite as much, which is the oral microbiome. So thinking about the oral microbiome, those ohms that colonize our mouth, there's an interaction that happens with both the host creating an environment that might preferentially select for specific microbes to colonize, while also the microbes influencing what that host environment looks like. So there's this feedback interaction loop that's kind of creating this community of microbes. And when that interaction is living stably and healthfully, we call that a eubiotic state. So a microbiome that's healthily living within the host and potentially even benefiting the host. However, when we get an unbalance of those microbes, potentially an overgrowth of a specific bacteria or fungus or a pathogens coming in, this can cause disease, and that disease-causing unbalanced microbiome is what we call a dysbiotic or a microbiome in dysbiosis. It can cause, in the case of the oral microbiome, it's been connected with a lot of different diseases, both in the mouth and outside of. So I want to talk about this broad influence of the oral microbiome on health. So I'm thinking about this oral microbiome influence on health through three different mechanisms. The first mechanism is kind of a local effect, and this is pretty self-explanatory. The microbes in your mouth are going to impact the health of your mouth. If you have a bad microbe that is going to predispose you to tooth decay, that can cause, or potentially gum disease, that can cause some oral issues and impact your mouth health. However, I want to also talk about the other way that the microbes in your mouth can find their way to other sites of the body and impact the microbiome and thus your health in other ways. So this is through colonizing other sites, and we're going to focus on specifically the gut and lung here in this talk. So going back to that tube in a tube analogy, that inner tube is all connected, starting at the mouth. And so constantly throughout the day, you are swallowing saliva. And in that saliva, you have microbial hitchhikers that are going to come along. And so they're going to pass through your stomach and potentially colonize your gut. And while most microbes won't make it past kind of that stomach acid, acidic environment of your stomach, they will still, some of them, colonize your gut. And so studies have shown that microbes that are present in your mouth do influence what is present in your gut. And it's also kind of that first route of exposure. If we have a bad defense system or a microbiome in our mouth that's going to allow for kind of some nasty bugs, those can eventually end up in our guts, potentially. So that's one way where our mouth microbiome influences our gut. But what about our lungs? So just like you're routinely swallowing your spit throughout the day, you actually are aspirating small amounts of your spit throughout the day as well. So aspirating or breathing it in. Think about when you've kind of been talking and you accidentally breathe in weird and you start coughing because it went down the wrong pipe. That's kind of an example of a large amount of aspiration of spit into the lungs. And just like with swallowing, some microbes are coming along, they're hitchhiking along in that saliva. And so depending on what microbes are in your mouth, you're changing your lung exposure. And while a healthy lung isn't going to have much bacteria in there, it's pretty good at clearing it out. Imagine how if you're giving your lung kind of easy, not very threatening bacteria to clear versus potentially pathogenic or really not very human body friendly bacteria that it's having to clear and combat routinely. And so the health of our mouth can really impact the health of our lungs directly through that colonization. But beyond those bugs being specifically present in certain spots, there are systemic health effects that we're seeing of our oral microbiome. And this can happen through the immune system. Our bodies are connected by this immune system. This diagram outlines where our immune system is throughout our body, really simplistically. And I want to draw your attention to this kind of green web that's going throughout this diagram of this body. This is kind of that lymphatic or that immune system highway, and it forms this connection between all of these different immune sites throughout your body. So it's a way that what's happening in your mouth and in your lungs and in your gut is connected through your whole body. So you could understand how what's happening in one immune environment could impact the rest of the body. And so to think about specifically what this looks like in the mouth, let's think about here at the top left of the diagram, how if you have potentially an infection or inflamed gums or an inflammatory microbiome, how this could produce bacterial products that are nasty or other immunoinflammatory host products that then go into circulation with the immune lymph communication highway. This then can impact other body sites such as your heart, causing various cardiovascular diseases, your lungs with pulmonary disease, or your liver or pancreas and causing metabolic diseases, or at least influencing your development of these diseases. Even things like your joints, rheumatoid arthritis is an inflammatory disease that has been directly connected with specific microbes in your mouth. So there are these big systemic effects. And I want to touch on an example of something quite relevant in our age of COVID on how the oral microbiome can influence lung infection. So as I mentioned, that having a dysbiotic or out of balance or a oral microbiome that potentially contains these nasty pathogens can increase maybe some of that immune signaling, can make your immune system almost a little stressed out chronically. And that can kind of open the door or make you more susceptible potentially to viral infections. So in COVID, if we can understand microbes that are potentially protective or harmful for acquiring COVID or developing severe disease with COVID or even influenza infection, we could potentially find mechanisms to protect people from hospitalization, for example, if they get sick. Additionally, a common thing after a viral lung infection is what's called a secondary infection. This is a bacterial infection that comes during or very shortly after you have a viral infection in your lungs. your immune system can kind of get this tunnel vision where it's really focusing on fighting a viral infection, and a bacteria can kind of fly under the radar a little bit. So you can imagine if you have some nasty bugs in your mouth or that are routinely appearing in your lung, they might fly under that radar during a viral infection and potentially cause other pneumonia or other diseases. So if we, again, can know ways to protect against that, we can improve lung health and reduce hospitalizations in these kinds of lung infections. So this oral microbiome really holds the key to a lot of very pressing health conditions that we're thinking about today. And lastly, I just want to bring back to this diagram at the very bottom and how what's exciting about the big influence that this oral microbiome has on the systemic health is that it is an easily accessible site. I mean, we can see our microbiome, we can change oral hygiene habits and directly influence our oral microbiome. So it's a really exciting opportunity to modify our microbiome both orally and potentially in other sites of the body through a lot of different exciting technologies. So now that we've addressed some foundations of what I want to talk about the microbiome and oral microbiome, I want to talk very briefly about some of my own work on the saliva microbiome to just give you a taste of some very basic methods and concepts when we're thinking about study. So when we're measuring the microbiome, a main question that we're asking is who's there? The who being microbes. What species are in the oral microbiome? How are they different in healthy people versus people with periodontal disease? Who's there? Well, how do we measure that? There's a lot of different ways, but a very common way is through sequencing, utilizing the genetic material, specifically the microbial genetic material in these systems. So an example of what a pipeline like this would look like. Let's say we collect a sample. Maybe it's an oral swab or a nasal swab. And we then isolate all of the DNA in that sample. So we get all of that genetic material. We can then use a very specific bacterial gene. So here with the microbes, we're only thinking about the bacteriome or the bacteria. But there's a specific gene called 16S rRNA gene that's highly conserved throughout most bacteria. And it has these hypervariable regions that can act kind of like a fingerprint that can tell us uniquely which species, which kind of genetic fingerprints are there, who's there. So we collect up all of these fingerprints, we run them through next generation sequencing, and then we get out all of this data about who's there. And this data analysis is really at the heart of the methods that we use for microbiome studies today. And I want to talk very briefly about that. So to illustrate a very simple version of what this would look like for an oral microbiome, I want to talk about some recent work that I've done looking at the saliva microbiome. So this is just a small study where we took 22 people and we asked them to give us 2 saliva samples. One that was just saliva in an empty tube, and another that was collected with a special preservative that was meant to preserve the genetic material in the sample. We then took those 2 samples and ran them through that 16S rRNA gene sequencing pipeline, that fingerprinting pipeline. And we got out all of that data, and then we figured out who's there, who's in that sample. And so here's one way that we can look at who's there. This is essentially the number of species, or what we call species equivalents, in the saliva microbiome. I put that in quotes because we're not actually growing and looking at specific bacteria. We're just looking at evidence that they're there, looking at their genetic material. So here, the number of species is marked on this graph between 50 and 200. And I've split it up in samples that were collected just saliva with no preservative here in blue, or those collected with preservative on the right in red. And samples that came from the same subject are connected with that gray bar. And so there's 2 main takeaways I want you to get from this graph. First is the variety and the range and the number of observed species that we see in these saliva microbiomes. I mentioned at the beginning that there's this feedback of the host environment kind of dictates what the microbial environment is like and vice versa. So these differences are potentially host dependent. Depending on oral hygiene habits, diet, how much sugar you're eating, can really impact what your microbiome, the species, the who's there, looks like. Second, I also want to note as part of the study is that the samples that were collected with preservative on average, had a higher number of observed species. So this shows an interesting conundrum that we have in this microbiome field, and that the way we collect samples influences how we're able to understand them and analyze them. So we have to think, really deeply about how are we collecting this and how is this giving us an actual picture of what's happening. Something that's kind of a tricky task even today when we've been studying this for a while. So this is looking at who's there. This is counting just the number of species. And we can look at, people with preservative have this species, people without preservative have this other species. But when you have 200 different species that you're looking at, listing all of the ones that differ between them can get quite complicated. So one way that we get around this is through statistics and bioinformatics to try and compare whole communities together. So I want to illustrate this with some mock communities. Let's say we have communities A, B, and C, here represented by 3 Petri dishes. And each point in these represents a species or a microbe that we found. So we identified who's there, and we're able to quantify the number of species. But how do we know which microbiomes are more similar to each other and which ones are different? Are C and B more similar to each other than they are to A? Well, we have different tools or algorithms to compare entire communities to each other and see how similar or different they are. And when we have, for example, 22 samples from different people, we can kind of understand how all of those look together. Again, looking at a whole community. And this is a really common technique used in microbiome samples. And so if you look into any microbiome study literature, most of them are going to include something similar to this, which we call here a principal coordinate plot or PCOA plot. Here, each point on this plot represents a single sample and the entire microbial community that we measured within that sample. Now, this is coming from that preservative, no preservative study, where the samples that are blue circles are those plain saliva samples collected just alone, and those in the red triangles are those collected with preservative. And samples coming from the same subject are again connected by a bar. And so how we can understand this graph comparing these whole communities is that points or communities, these samples that are closer together, are more similar. So things more similar will group together, whereas things that are farther apart on this graph, say this one down here and this one up here, are more different, more dissimilar. And so we can look at these grouping patterns and see how they relate to each other to try and understand overall how these communities differ between each other. And so here, the takeaway from this, well, it's quite messy, potentially not the most clear illustration, is that individuals are more similar to themselves in general, when even despite the differences in collection methods. This varies from person to person. If you look at this individual that I'm highlighting here, their points are almost exactly identical. There's really no difference when collected with preservative versus without. But if we look at this person down here at the bottom of the graph, they're quite different between each other, between the preservative and non-preservative. So this can give us a lot of different interesting data to try and extrapolate. Maybe there are microbes that associate with different areas of the graph. But essentially, when you see these data, I want to press upon you that this is looking at full communities and how these communities are similar or different. And again, this is going to be an extremely common thing that you see throughout the microbiome literature. So this was a lightning fast look at the oral microbiome, some general microbiome thoughts. There's more to come within this, but I want to give you just some concluding thoughts that I want you to dwell on after this. So the microbiome consists of more than just bacteria and exists where our bodies have this outside exposure, the tube within a tube. The oral microbiome impacts health beyond the mouth, colonizing other body sites, making products, and triggering the immune system. And this impacts our health. So the microbiome is also studied through sequencing primarily. This 16S sequencing targets a fingerprint gene to tell us who is there. It tells us the microbes within the community. And how we collect and view body sites can change how we view the microbiome there. This is illustrated by that saliva sample study that I showed you. And lastly, bioinformatics is at the core of how we understand this work. I didn't dive too much into the bioinformatics, but know that a lot of very smart computational biologists came before me to develop those tools for me to be able to show you those graphs of that data. So it's a really exciting and still growing field of understanding this sequence. And so lastly, we just want to acknowledge the people who I work in the Martin Blaser lab at Rutgers and the Center for Advanced Biotechnology and Medicine. And I have some contact information and socials down at the bottom. I am on Science Twitter, but I don't tweet much. But if you want to hear about some sporadic microbiome thoughts, you can follow me there.
Awesome. Thank you so much, Dr. Armstrong. It was so great to learn about our oral microbiome. So now we'll move into the Q&A because we got a lot of questions. So first, could you discuss superbugs in the relationship to the microbiome?
I really think the microbiome is a tool that we can use to combat them. If we think about how, if you have a eubiotic, one of those healthy, thriving, diverse microbiome communities, let's say on your skin, that could potentially protect you from, MRSA or VRSA, one of those staph infections that are really antibiotic resistant from taking hold. So it's kind of protective against superbugs. But also we know that we're finding certain microbes within, for example, the skin microbiome that compete really well with a staph infection that's resistant to microbes. So it could definitely be utilized to, outcompete infections and help cure our infections. So that's often what I think of.
To address the relationship between the changes in the oral microbiota from infancy and how that could relate to diseases in the future.
I talked just on the oral microbiome, but really I think a lot of the research is, especially in kind of the lab group I'm a part of, looks at the gut microbiome and how the colonizers, the bugs that kind of start your microbiome. So from birth, those first founders really impact who comes next, and that impacts your overall microbiome going forward. And what we see is that infants who are given a lot of antibiotics, or children under 2 who maybe are given antibiotics for like recurrent ear infections, that changes their microbiome, and we see connections with that altered microbiome and other diseases such as diabetes and allergy. So that infant early microbiome colonization it starts immediately, and those impacts kind of compound, and we are seeing that it truly does impact health.
Does the microbiome have any impact on our mental health?
I showed that diagram of how our immune system is like thoroughly connected throughout our body. What I didn't show is a similarly complicated map of also our nerves throughout our body. So we sometimes call it the second brain, this network of nerves that are around our gut, that help our gut move, but they are connected back to the brain, the immune system, the circulatory system. All of this is influenced by what's happening in our microbiomes. And that definitely can influence health. We see in some mouse models, lower levels or higher levels of anxiety, depending on certain microbes or just microbially produced products that are in circulation. And there's some people looking at, autism, and how, people with autism tend to have some GI upset, or, a subset of people with autism. And they're looking at fecal transplants as a way to both alleviate those symptoms, but also potentially improve, behavior and functioning there. So definitely there is what we call a gut-brain connection that can happen through a lot of different mechanisms.
So fascinating. There's always more to learn. It's the best part about microbiology. So are there any microbes that you can detect in the microbiota that specifically will give you indication that this person may develop a digestive tract cancer or Alzheimer's or any other type of diseases?
There's some things, that suggest a potential predisposition. Some things that might suggest you are more likely to develop a condition like, rheumatoid arthritis, some inflammatory condition, but the microbes aren't the only thing acting there. Your body is kind of taking what it's given. So the microbes are just one part of this complex system of disease. So there's no for sure when we're thinking about microbes.
talk about a few things we can look forward to in the future in terms of microbiome research and curing obesity or diabetes or any other type of health.
What I think with disease is we'll see a lot of like adjuvant treatments or treatments that go along with how we're currently treating diseases. So this could be, and we're already seeing some of this, microbes or like a probiotic that you take along with like a cancer med, and that helps alleviate the really nasty GI symptoms that you get from that cancer medicine. Or we're seeing that microbes kind of can influence the progression of disease. So if we can find ways to really have direct, specific microbial targets or microbes that we can use along with our current medicine, I think we could see fewer side effects or more effective treatments in the future. What I don't think we'll see is just everyone taking, probiotics or like a really direct microbiome targeted therapy. I think where the real power of it is going to be in these kind of co-treatments, if you will.
So thank you everyone for joining us. I actually just very quickly want to mention our sponsors for the Teacher Enrichment Program, which are the major sponsors include the Benedum Foundation, the Virginia Department of Higher Education, Illumina, BAE Systems, Claude Charitable Foundation, Akamai Technologies, Aerojet Rocketdyne, Equitrans Midstream, Jacobs Engineering, Lockheed Martin, and TC Energy Foundation. So thank you all for joining us.